So hi, such a pleasure to be here. I have such awesome, vivid memories of actually being in Grace Auditorium and there with everyone. So it's very difficult not to be hanging out at the garden and all of that, but it's still fabulous to have so many of you. So thank you to the organizers for putting this together despite these difficult times. So what I'd like to talk to you about today is our effort in trying to dissect the disease circuitry of the brain. So the overall goal is to understand the molecular basis of human disease. And the goal is to develop new target genes, new therapeutics, personalized medicine. The challenge, of course, is that when you start looking deep down as to what actually goes on in these regions, what you're finding is that the vast majority of these loci are in fact non-coding. So that makes interpretation very difficult because we can't find the target gene, the causal variant, the cell type of action, the relevant pathways and the mechanism. So the approach that we take in my lab to address this challenge is that we start, of course, with disease genetics, with common and rare variants, and then we systematically profile RNA transcriptome in both healthy and disease samples. We integrate all the data to predict driver genes, regions, and cell types, and then we validate the predictions in both human cells and in mouse models. The goal is to bridge the gap, the huge gap between genetics and disease, with thousands of very weak effect variants into smaller parts where these genetic variants are expected to have bigger effects, and each of these regulatory regions, bigger effects on the genes, and so on and so forth, on the way to disease. And of course, this is challenging because we want to understand both causality and feedback from the disease. Ultimately, what we're hoping to do is basically use that to predict driver genes that are either positively or negatively correlated with the disease and that are driven by genetic variation. And this is an example in bulk brain samples for Alzheimer's disease, a method developed by Youngjin Park in my group. The other, of course, success has been that if you look at dozens of tissues and dozens of traits, you find this very nice correspondence that allows you to sort of guess which tissue every trait might be acting in. And we've scaled this to thousands of samples, hundreds of different tissue types, and painted very complex pictures of how these disease variants are localizing into and enriching into different tissues. And in this particular case, you can see brain here contains schizophrenia, neuroticism, worry, depression, as well as educational attainment, intelligence, and going to calls for Harvard conferences. Whereas there's on the opposite side, Alzheimer's disease is in fact localizing with immune traits, and that's something that we saw early on. And the reason for this is that if you actually sort brain samples into microglia versus neurons versus other glial cells, you see that the genetic signal appears to be highly localized in specifically microglia. So that's what motivated us to start now looking at single cell dissection of brain disorders. And we started out with Alzheimer's disease. What we found in a paper that was published about a year ago, single cell transcriptional analysis of Alzheimer's disease, is that we can actually start profiling thousands of cells across dozens of individuals. In this particular case, 84,000 cells, 48 individuals, non-AD, mild AD, severe AD, and then start classifying where did each of the cells come from and what are the molecular changes that we see. And the results were quite striking. The first thing that we saw is that even though microglia appear to be the dominant driver from the genetic signal, every major cell type saw both AD and non-AD associated subclusters after clustering each of those. The other quite striking thing is that female subjects showed a systematic enrichment for the more disease-associated subset of those cells. We also found that the early changes in AD were in fact extremely highly tissue-specific, whereas the late changes are actually quite common and more associated with stress response. We found that the differences between men and women were quite high to begin with, 3,000 genes differentially expressed in neurons, but became even larger in the context of AD. And in particular, we found that men upregulated oligodendrocyte pathways, but women did not. And this led us to hypothesize that in fact there would be myelination differences between men and women, and indeed, women lost a lot more myelin than the men, consistent with what we found in pathology images. We have since scaled up by multiplexing and also profiling more than 1,000 samples. We've basically now started efforts in Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in the context of AD, schizophrenia, bipolar, Down syndrome, autism, depression, suicide, PTSD, and aging. In each case, profiling about 24 cases, 24 controls, totaling 1,500 individuals, looking at all of the major cell types, and a total of 7.5 million cells across all this, and up to 7 brain regions across both RNA and attack. So here's a summary of the data that we've generated so far. This is, the different traits that we're studying, the region, and then the technology, and then the tally at the bottom. We have 900 cases, 700 controls, 200 trillion cells, 7.5 million, 200 trillion reads, 7.5 million cells. The multiplexing method that we're using allows us to now start profiling multiple traits in the same batch, enabling us to now correct batch effects between different traits in a very nice way. And that allows us to now start looking at systematically what are the differences that we're finding in each of these traits. So if you look at schizophrenia and bipolar disorder, you basically find that the differences are very highly localized in axon guidance, synaptic vesicle cycle, regulation of neuron projection pathways. And then these are very different between different types of cells. We're also finding that the genetic signal for schizophrenia appears to be localizing primarily in inhibitory neurons, even though the changes are found across both excitatory and inhibitory neurons. And these are the genes that are differentially changing, and these are the pathways that are differentially changing. We also looked at amyotrophic lateral sclerosis, so ALS, Lou Gehrig's disease. You basically see here that ALS versus FTD, which are sharing a lot of pathology, but in different regions of the brain, appear to be, in fact, going in opposite directions in the motor cortex. And in fact, the genes that are associated with ALS are painting a very rich picture of both endoplasmic reticulum and autophagy as some of the top pathways. And surprisingly, they appear to be acting in astrocytes and oligodendrocytes rather than neurons directly. In Huntington's disease, we're basically finding that the single cell data of the striatum, of the striatum specifically, is looking at differences between these indirect pathway spiny projection neurons and these direct pathway spiny projection neurons, which appear to be merging in the context of HD. So they are extremely distinct in normal samples, but in fact lose their identity, giving us some hints as to what might actually be going on. We've also scaled this across multiple regions. In the context of AD, for example, we've profiled 7 different regions across a total of nearly 400 samples. Across prefrontal cortex, midtemporal cortex, angular gyrus, entorhinal cortex, thalamus, hippocampus, and mammillary body, each in 48 or more individuals. These are the clusters that we obtain. These are the cell type annotations for those clusters. And the striking thing is that if you look at neurofibrillary tangle pathology versus neuritic plaques, you see that they're affecting different regions of the brain extremely differently, enabling us again to focus on this dramatic alteration between these regions. But we can go into sub-region resolution as well. So looking at the hippocampus, for example, you see this dentate gyrus, region CA1, CA2, CA3, CA4, and the subiculum are in fact very easily distinguished from the single-cell RNA-seq data without even doing spatial transcriptomics. So from the cell type identity, there is actually sufficient resolution in the dataset to actually distinguish these dramatically different cells from their gene expression profiles alone. And you can see here how the samples are in fact varying across individuals. And you can also see that the dentate gyrus, for example, is the least predictive of AD, but CA1 appears to be the most predictive of AD, enabling us to focus on 2 sub-regions that are the most informative. We can also ask what are the genes that are differentially active in each of the sub-regions now of the hippocampus. And you can see that there's actually quite a lot of variation in the specific genes that are altered and the specific pathways that are enriched in different subtypes. We're also able to focus on individual subtypes of cells. So for example, if you look at microglia, we now have more than 150,000 microglial cells across all these samples. We're able to now distinguish different states of microglia. You can distinguish, mitotic, phagosome, antigen processing, anti-inflammatory, enriched microglia. These are the pathways that are enriched in each of these subclusters of microglia. And we're also able to recognize these brain-associated macrophages, which are, again, circulating blood cell type. We're also now able to look at epigenomic variation in the context of genetic variation and transcriptional variation and start integrating single-cell attack and single-cell RNA data. This is basically just a glimpse of how the attack data maps across different cell types. We can look at variation across individuals in attack peaks and correlate that with variation in RNA reads and start mapping non-coding regions to their target genes in cell type specific links. Here's some examples where we can see coordinated changes in both the RNA signal and the attack signal in the context of disease. Here's an example, for example, in neuronal cells, astrocytes, microglia, oligodendrocytes. ApoE, for example, is increasing in expression in RNA, and indeed you see its accessibility increasing in single-cell attack. RLPB1 decreases in expression in AD, and indeed accessibility decreases, and so on and so forth. You can also see these coordinated changes across many, many different genes. So on the x-axis here is the change in RNA expression from single-cell RNA in that cell type. And on the y-axis, the accessibility change for the same region of the same gene. And you can see there's some genes that change coordinately, whereas others can change in expression even when their accessibility is not changing. We can, of course, do the single-cell attack across multiple regions, and we can now start studying region-specific differences. We've also started fine mapping regions of genetic association using the single-cell attack data. So when we do that, you see, again, what we would have expected, that the microglial cells are, in fact, hugely enriched for AD-GWAS, whereas all the other cells are depleted. And across different brain-associated traits, you see that, for example, for psychiatric traits like schizophrenia, neuroticism, and IBD, you see these enrichment for excitatory and inhibitory neurons, whereas for Alzheimer's and other immune traits, you see this enrichment instead in the microglial cells. We can also start, of course, because we have AD and non-AD individuals that we have single-cell attack data from, we can actually start looking for changes that are happening in the context of AD. And in this particular case, you see this dramatic increase in accessibility for the risk haplotype in non-AD individuals, but no such change in the AD individuals. So this is kind of interesting. It basically says that the risk allele has an effect which is trait conditional. So that basically suggests that a G by E interaction, which might indicate why EQTLs are not generally found to be meaningful in the control samples, maybe you have to actually go into the disease setting in order to find these significant differences. We've also exploited the large number of samples to call EQTLs at the single cell level. So we've basically processed these 480 samples in prefrontal cortex alone with associated genotype, and we've now got cell type specific EQTLs. And we can take GWAS from any trait associated with brain and start asking in what cell type is this particular variant associated with that particular gene changing the expression of that gene in a cell type specific way. And you can see some genes acting in, some variants acting in oligodendrocytes, while other variants are acting in excitatory and inhibitory neurons to either increase or decrease expression. And this changes dramatically from trait to trait. If you look at bipolar or major depressive disorder or Alzheimer's disease, you see a different set of traits. We can also actually carry out causal mediation analysis to look at which of these genes actually capture the heritability of that trait through their gene expression profiles and start predicting both the effect, the direction, and the cell type where each of these genes is acting. We can also look at the sequence information that we get from single-cell RNA-seq data to infer mosaic mutations that are occurring during the development of the human brain and are in fact impacting only a subset of the cells in our assays. When we do that with SmartSeq data, we're basically finding these disease-associated neurons and disease-associated oligodendrocytes show a dramatically higher number of mutations than non-disease-associated cells, and glia in general that are post-mitotic, that are pre-mitotic, are in fact showing more mutations than post-mitotic cells like neurons. We're also finding differences in the burden of somatic mutations between disease and non-disease individuals and between male and female subjects. We're also finding that these burden increase is actually cell type specific, so a different set of pathways is enriched in excitatory neurons versus oligodendrocytes. And here's one example of a clonal hit, which is affecting multiple oligodendrocytes and multiple astrocytes from the same individual. We've now started systematically validating our findings in collaboration with Kevin Egan and Kristen Brennan, with Maria Kusi, Michael Goodbrod, and Kevin Smith, to basically take the list of predicted driver genes and the cell types where we're predicting them to be acting, and how they're acting and how they're associated with GWAS, and basically construct these programmable assays to basically differentiate iPSCs with Cas9 or dCas9 with activating or repressive domain into the different neuronal and glial cell types, and then start asking for their systematic effects. We can actually see some of these remarkable changes that are happening at the cellular level. We can actually test assays like calcium signaling in 384 well plates, and we can also carry out massively parallel reporter assays and insert actually some of those assays into the mice, in this case in collaboration with Andreas Penning in the context of the Cure Alzheimer's Fund Consortium. Incredible set of individuals to thank, awesome collaboration, particularly with Li Wei Tai and her lab. Many of these folks have now gone off to start their own faculty positions, so please join our lab. We're looking for postdocs, and I'll stop there and take any questions.
Okay, thank you, Manoli, very much. I'll start out with Ekta Khurana. For what fraction of genes do you find consistent trends between single-cell ATAC-seq and single-cell RNA-seq data?
Oh gosh, we haven't quantified it, but we're initially focusing on just the cells, the genes that show agreement. And in the plot that I showed, you could see that all of these arrows were particularly pronounced for the strongest effect genes, whereas in fact, the weaker effects were less consistent. So as you go to the most pronounced effects in both RNA and ATAC, then you actually generally see agreement.
Okay, next is Gloria Novakova, PhD student. How many EQTLs do you identify in single-cell microglia? How do they compare to the recently published bulk EQTL data?
So bulk EQTL shows the strongest agreement with excitatory neurons and inhibitory neurons. Microglia are highly underrepresented in the bulk QTL analysis. So, and I don't have global statistics about the total agreement, but we do capture the vast majority of the neuronal ones in the bulk, but when it comes to the glia, we really don't capture those. I don't have exact statistics.
Next, Anna Mathiodecki she's a PhD student in EMBL. For this study, you look into multiple individuals from a single cell perspective. Do you observe high among individual variability in terms of the numbers and identity of clusters? And if yes, how do you account for that?
Actually, surprisingly, no. We've had to do very little batch correction. There are methods out there for batch correction. We haven't had to use them because we actually find very, very good agreement in the cell types between individuals. There were some examples, for example, between regions. The mammillary body showed slightly different clusters from the other cell types. For those, we don't know if it's biological or technical. We expect it to be mostly biological because of the agreement that we see in many of the other samples. So that helps.
Do you see strong colocalization of GWAS hits with EQTLs, and is one particular neurological disorder showing a stronger overlap of GWAS hits to EQTLs?
Surprisingly, no. EQTLs do not show this global enrichment, but as I showed in my example, this might be in great part due to the fact that they are condition-specific EQTLs. In other words, these EQTLs only become EQTLs in specific contexts, and we've seen this in multiple published papers, where upon activation, the EQTL will have its effect. In the example that I showed, it's only the case of Alzheimer's that you actually see this very strong EQTL effect. And we saw the same thing with the FTO locus. It's only the context of pre-adipocytes that the EQTL was active. In the whole tissue, it wasn't apparent. So I think that's part of the reason why the field is not finding such a strong agreement between EQTL and GWAS, mostly because a lot of these effects are condition-specific.
Okay, we'll go ahead with, there's lots of
interest. We have one more
interest.
minute, come on.
That's right, we'll keep going with a couple more questions. Dingwen Su, PhD student, how is data normalized across multiple samples and tissues?
So we do a lot of normalization, usually in a per project kind of way. Normalizing across the 7 million cells has been kind of daunting and challenging, although we've done some of that too, and there's a lot of sort of streaming methods that we've developed for that, and I'm happy to talk a lot more about that at the break.
Okay. Javi Mohammed, a postdoc, you've talked about zinc finger proteins and Hox oh no, that's an old question.
Yeah.
Strike
we
that
did
from
them
the
all.
record.
That's awesome.
Can you please talk about your effector domain screen? That's the one thing you didn't talk about. Okay, next, anonymous attendee. What is the distribution of the ethnicity of the patients and normal human samples? Are they
Mostly European. So in the ROS-MAP cohort, we have some non-European samples, but it's unfortunately biased by where the genetics is, where the samples are, etc. So we are not doing a very good job about sort of cross-ethnic. We have some projects underway that are sort of moving in that direction, but no results have shown that yet.
Okay, and I'll ask one last question related to Alzheimer's and such a strong signal that you see in the microglia. Given your view of all the cell types that you've queried, what is your view on the genetic effects that are happening in the neurons? And is it at all, is there any evidence for a primary effect, or is your current speculation that the effect on neurodegeneration is entirely secondary through the microglia?
So I wouldn't say that it's entirely secondary. To a great part, we see this in huge enrichment for microglia, but we do see QTL effects in other cell types as well. So I would not discount them. The other thing to realize is that there's a lot of cross cell type interaction and crosstalk. So even if an effect is primarily acting in the microglia, then sort of you can actually observe its effect almost directly in other cell types. And there's a lot of sort of disease-specific effects that are sort of mediated through that. I would say stay tuned. I would not discount genetic effects in other cell types. It's just that the vast majority are enriched in microglia. I would estimate slightly more than half will be the final tally. And when we're doing our QTL analysis, we're finding that maybe 60 to 70% are in microglia, and another 30 to 40% are in other cell types as QTL correct.
Great. There's individuals here, Thomas Lugunas, who's emphasizing how interested they are in getting their hands on the data, which is always a good sign. And Manolis, thank you for a great talk. I'm going to leave this in the chat box. There's one other question for
great. We'll
you to get
after. As soon as we're done with the QC. I mean, all of these data was generated in less than a year. So it's, we're still cracking through making sure that all of the cell types are correct, etc. So stay tuned. We're also looking for people to help us build all the websites for this. So apply to the lab, help us get the data out.
Okay, great. So we're going to move forward with the last talk of this first half.